Hey guys, Kriben Govender from the Gut Health Gurus podcast. I've got a background in food science. My colleague Jane Shadrack, who's all things psychology, is unfortunately not able to join us today. He's got another appointment. But I've got an amazing guest for you today. It is if you recall, I did interview Dr. Jack Cruz a couple of weeks ago, and today I've got someone who I equally follow very closely and all his work, especially around deuterium. And I have the pleasure of introducing Dr. Laszlo Boros. Dr. Laszlo, welcome to the show.
Well, thank you very much for inviting me. It's an honor.
And typically when we start our shows, I like to set the scene for our audience, and to introduce our guests. And what better way to introduce a guest than from the horse's mouth? So, who is Dr. Laszlo Boros?
So I do teach biochemistry at UCLA, and this is the University of California, Los Angeles. And I'm a physician. I'm a doctor by training. I got my medical diploma in 1987 from Szeged, Hungary. I grew up in a smaller town in Hungary, and then I went to practice medicine for a short time. Then I left for Germany and then to the United States, and I was appointed in 1998 as one of the faculty members of the University of California. And I've been working for the Department of Pediatrics, but my work was mostly research related and studying metabolism using glucose molecules that actually have a special tag on it. We call them 13C tagged glucose, and I followed the fate of glucose in many disease scenarios, in many disease processes. And then around 2002, I came across with deuterium, working with Dr. Gabor Somlyai, who is my dear colleague from Hungary, and we have worked on various clinical applications of deuterium research, and then now we are delivering deuterium physiology or deuterium biochemistry as part of our education process at UCLA, and we also have a clinic where we teach how to practice deuterium biochemistry or deuterium awareness for your health in Los Angeles. In Brentwood, we have a clinic called DD Centers, and so that we are getting into the clinical or the health applications of the topic.
Sure. And you mentioned Dr. Gabor Somlyai, and I was fortunate enough to have a conversation with Dr. Gabor quite a few weeks ago, and I'm just fascinated. How did you guys actually meet?
It's a funny story, I have to admit. I heard about deuterium first, or deuterium depleted water, from a friend in Los Angeles who is actually involved in freighter shipping. So he's just not a scientist from any angle. Yet he was telling me about this water, and I was kind of skeptic, like anybody else would be. And he wanted me to do a study, and eventually, after the right arrangements, we made a study design with Dr. Gabor Somlyai, and sure enough, deuterium depleted water had major basic fundamental effects on cellular metabolism, which we studied with glucose. As I mentioned before, we do have glucose tracer type experiments for precise biochemistry explorations. And sure enough, there's a dose-dependent pharmaceutical metabolism. And for a few years, I worked on the topic. Yet I got a phone call from the National Cancer Institute, from the urology branch, where they had a patient who had a mutation in the mitochondria of an enzyme that resulting in very aggressive cancer. And so that was the biochemist, this International Institute, and we came up with strong data indicating that actually this water, which we call metabolic water, and is produced in mitochondria, is very critical to maintain cellular health. As long as there's no deuterium or heavy hydrogen isotope in it. And from then on, we kept publishing, and we have been continuously delivering this information and knowledge to doctors, colleagues, everybody who's interested in the topic, including your previous guests like Dr. Jack Cruz and Dr. Gabor Somlyai. And now it's Dr. Dominic De Agostino. I just sent you a post. He's also a very good friend and colleague of us. So it's becoming more of very hot, very interesting, very new, yet very fundamental.
I think we dropped off for a bit there in the last section. So I think I caught the part about Dom Agostino and your new work that you're doing.
Yes, indeed. We are interpreting ketosis, ketogenic diet, and effect of light and melatonin, which is a deuterium depleting hormone in the sense that it helps during sleep, function related to water production. And so this is more like it's getting a very fundamental, very basic, yet very interesting and new topic for a group of investigators, including myself and obviously many of my very highly esteemed colleagues.
Sure. And look, I think it's a good opportunity now to just take a step back and define to our audience, who are very much a lay audience, about some of these terminologies that you introduced onto the podcast. So the first thing is, how about we define deuterium, which is, I think it is a hot topic. It's going to be even more prominent in the coming years. I think we're very much at that early adopter stage with people learning about deuterium. So what is the lay, I guess, explanation of deuterium?
Well, the lamest way to say this is probably a twice as heavy cricket ball. Am I saying it right? You guys play cricket.
Yeah, absolutely.
Is that right? Yeah, or a twice as heavy transfer of energy in cells by definition is actually stable heavy isotope of hydrogen. Hydrogen is the first element of our periodic table, and it's the most active in biological processes in transferring energy. And helium, it's twice as heavy counterpart called stable isotope. It has one proton and a neutron, while hydrogen only has one proton, the nucleus of the hydrogen atom. Obviously, there's an electron around, but yet it's like we destroy the game, and that's what deuterium does. It unfortunately
Sorry, Laszlo. I'll pause you there for a sec. I think we're going to have a lot of issues with the connection. So what I might do is I might just stop the video and we'll just do audio, because I have a feeling there's a problem with the internet connection. You still there?
Audio?
Yes. I think we'll do audio only, because I think the internet connection is not great.
Okay.
So how about you?
Because we don't have to have a picture, is that right?
Yes, we can still talk, but I think it's lagging. Yes. Yep.
Yeah,
I think
I did turn it off.
Yes, awesome. I think it'll be easier with the internet connection.
Okay.
So how about we just take a little step back, because I think it was just really breaking up. It was very difficult to understand. So what we'll do is we'll just take a couple of steps backwards. So how about we start again with the definition of what deuterium is?
Yes. So in very, very late terms, deuterium is like a heavy cricket ball or a heavy football that destroys the game practically. And this is what deuterium does in our cells when it gets into proteins and spinning proteins, working proteins, and particular parts of mitochondria, for example, these nanomotors that produce ATP. It's by definition, it's a heavy isotope of hydrogen. It has a proton and a neutron in its nucleus, while hydrogen only has one proton. And this extra neutron makes deuterium twice as heavy. And as in any biological process, the transfer of atoms or transfer of energy in the form of hydrogen slows down when it becomes a deuterium, and that would actually devastate some of our basic fundamental cellular functions. And that's in lay terms, that's probably how I could summarize the best what deuterium is.
Sure. So it's an isotope or a form of hydrogen. It's heavier than typical hydrogen, and it can interfere with the biological processes within the body.
Well, indeed, and it's twice as heavy, so it's significantly heavier than hydrogen, and this is why it makes it such a bad heavy isotope, because there is no other atom or other biological constituent or other biological component in the atomic scale that has such a dramatic 100% increase or twice as heavy characteristics when it comes to biochemistry and physiology. For example, carbon, which has 12 particles in the nuclei, it also has a stable isotope, which we call 13C, but it's only some 10% heavier, somewhere over 10, close to 10% heavier than its a 12C counterpart. So deuterium is bad because it's much heavier than its isotope counterpart, yet it would participate or try to participate in every chemical reaction that hydrogen would. So it's really a large increase in isotope effects, and this is why deuterium has such obvious and very prominent biological and health-related devastating effects.
So what are some of these devastating effects that having excess deuterium in the body can elicit?
So the most difficult part where deuterium can be processed is the mitochondria. Mitochondria is our little cell organelle that produce water and energy from the oxygen of air and from the hydrogen of food that we burn and oxidize in our cells. As we know, the result of this is carbon dioxide, which we breathe out, and also water, which we don't see much of it because it's recycled in a biochemical chain of reactions called the Krebs-Sanger cycle. But truly, hydrogen is very active in mitochondria, and more importantly, it actually drives or powers protein nanomotors, which produce ATP or energy for our cells. And these nanomotors are spinning at high velocity, about 9,000 at maximum speed, about 9,000 rotations per minute. And at twice as heavy hydrogen isotope, you can imagine what it does to a very delicate protein system moving at that speed. It makes it unbalanced, it starts wobbling, and after all, it breaks. So deuterium compromises our our mitochondrial energy production significantly, and it also slows down many other processes in cells that relate to biochemistry, but it also alters the structure of proteins and also the structure of DNA, because hydrogen bonds are very important in the holding together the two helices of DNA. And when these helices are bound by deuterium, DNA repair enzymes can go through. It's like when you mowing your lawn, and there is a big chunk of like grass somewhere, or there's some kind of a physical barrier, and you have to kind of go around. So deuterium physically damages cellular components that are necessary for energy production and also for healthy functions, and for that reason mitochondria is the prime, and peroxisomes, which also a place in our cells or cell organelle that produces water from dissolved oxygen and oxidizing fat, or burning fat. Those are cell organelles that are very sensitive structurally and functionally to the effects of deuterium.
So peroxisomes, are those akin to mitochondria, so small little organelles inside the cell?
Yeah, so mitochondria uses elementary oxygen, or just oxygen, carried by hemoglobin. It's a chemically very active oxygen because it is looking for a kind of a partner or counterpart, and deuterium and oxygen react very quickly, very fast. You probably remember from school, the exploding gas, oxygen and hydrogen mixture makes water very quickly. And practically the reaction is so quick that it sounds like an explosion. And that's what happens in the mitochondria in a very controlled fashion, obviously.
Sure.
And peroxisome, on the other hand, they use molecular oxygen, which is O2, which is in air. It does not require hemoglobin for carrying, for that matter. And not necessarily. For that reason, peroxisome can use this molecular oxygen and hydrogen from fat mostly to produce peroxide, hydrogen peroxide first, which is very rapidly, very quickly broken down into metabolic water, or water in our cells, by the enzyme called catalase. And this is the enzyme, for example, which melatonin in mitochondria and also in peroxisome would activate. So it seems that after all, it's all about deuterium and hydrogen capturing by our oxygen in either form, molecular or elementary form. Yet both organelles deliver metabolic water, which will have certain biochemical roles, very fundamental biochemical roles. For example, running the TCA cycle through recycling this water, or even becoming part of DNA by various, for example, gluconeogenesis, or pentose or 5-carbon sugar synthesis. And it seems that they are all connected, they are all linked, and they all originate from this water production process in mitochondria and peroxisomes.
So to give the audience an indication of what quantity of water as a percentage is produced by peroxisomes and mitochondria?
Yeah, so this is actually a very dramatic and large number, in the sense that actually we produce our mitochondria, and average human being produces about 7,200 liters of metabolic water a day, which is immediately recycled. And this is equivalent with the volume of blood we circulate. So it's not that surprising if you work with a physiologist or cardiologist, just to see how much work your heart performs as far as circulating blood through your body. It's practically 7.2 cubic meter of water getting produced and recycled in the form of protons
Wow.
each day in an average human being. And if you look at an average lifespan, it's about 700, somewhere around 700,000 cubic meter of water, which, for example, it's almost like a river flow, meaning that it's very significant amount of protons and water that we produce and recycle. And this is just to run the system. It does not mean that it's net production. It means that it's recycling the same protons and with new oxygen and new carbons, but it's practically just a more like your car's cooling water, practically just recycled so many times through the engine, and not necessarily a huge volume yet because of its flux, because of its recycling, because of its chemical activity. By the end of the day, you circulate about 7,200 liters of this metabolic water on an average day with like
Wow.
moderate activities.
That is a staggering number of the sheer amount of water per day. I'm absolutely stunned.
Well, and you recycle it. And if you look in your physiology book, there are other like shocking numbers, like for example, your kidney produces about 800 liters of primary filtrate a day, which are recycled back into your circulation. Your heart pumps about 7,200 liters of blood throughout your body a day. And indeed, your mitochondria produce and recycle about the same amount, 7,200 liters of water, which has to be deuterium-free to function properly. So it cannot have this semi-heavy or heavy water that we know from atomic like physics, in the sense that, heavy water is so different in its physical characteristics that they use heavy water to control atomic reactions in atomic power plants. So it is a very fundamental and a very critical knowledge, but it's never been applied to medicine until Dr. Gabor Somlyai started working on some cancer models and treatments with deuterium depleted water, and sure enough, the observed effects were quite dramatic in cell culture and also in humans, as far as their well-being is once their deuterium level is controlled and brought down to the physiological level.
Absolutely. Now, let's set the level of deuterium that's normal versus something that's elevated. From an environmental perspective, if we take tap water from Hungary, for instance, what's a typical number in ppm that it should be?
So we measure deuterium by parts per million, and that's just a very simple number. It would tell you how many deuterium there is in and amongst a million hydrogen atoms. In Budapest, drinking water has about 100, somewhere around 146, 150 parts per million in drinking water, and this is very typical of like a Middle European city. Usually coastlines have a little bit higher deuterium contents, because deuterium comes from the ocean after all, in the form of rains and vapors, because the oceans are the largest bodies of water in our planet and contain the most and the highest concentration of deuterium in water or open water surfaces. And because of temperatures, this evaporation and evaporation of deuterium from oceanic water, that after in the form of rain becomes part of crop and part of the vegetation, has shown certain kinetics and certain patterns based on temperature, wind, wind direction, speed, and so on. So it's this physical kind of fractionation or discrimination of deuterium because it's heavy weight, depend on physical processes, yet biological systems try to filter it completely out using these nanomotors, these spinning proteins that I mentioned a few minutes ago. And after all, when you look at ancient times, and probably this is how I should answer your question, back in the Paleolithic time, where actually humans and all like warm-blooded species adapted to the environment. Like if we look back 15 to 20,000 years ago, deuterium was in vapor by 10, 15 ppm lower.
Wow.
So meaning that if you drank water from a river 15,000, 20,000 years ago, instead of 150 ppm, it only had maybe 140 or 135 ppm deuterium. And that's what we consider as drinking deuterium from external extracellular water sources, external water. Then it should be in that range, below 130 ppm. We think that probably going back to like 100,000 years when most of the warm-blooded species have evolved in or have adapted to the environment, that's probably in the range of 120 ppm, and that's what we consider biologically relevant. And this is why we believe that depleting deuterium back to those levels benefit, after all, human health, and to that matter, all warm-blooded species. And again, it doesn't seem like a big difference, but because of the velocity and because of the transfer of protons in our system, this small difference goes very far as how often proteins and DNA needs to be repaired, or deuterium needs to be removed from our system simply because of the kinetics of protons and deuterium in biological processes.
And the burning question I have is why has the deuterium level increased from those Paleolithic times to the modern era?
It's by temperature. So if you warm up the ocean water, just like from ice ages, if you consider, well, it's very kind of easy for you guys because actually these studies were done in New Zealand. So physicists, they captured volcanic ash 15,000 years ago that was actually part of a volcanic eruption, and because New Zealand is a relatively small island, it's always covered by oceanic vapor. And when this ash, this explosion captured the oceanic water vapor, and they measured deuterium contents, they were actually 10, 15 ppm lower. Wow. So we know that these are actually, this is hardcore data in the sense that we do publish. I published about 100 papers in the medical literature, peer-reviewed papers in the medical literature, and we also look at peer-reviewed papers when we interpret deuterium-related findings, and this is just like part of it. So the measurements, it's not a guessing game. The measurements show that ancient times or Paleolithic times, deuterium was significantly lower in the environment. And the result of that is because of the lower oceanic temperatures. Once we went through geographical and also environmental changes because of the sun's earth distance and activities and the sun's activity, and there is a climate change, which we believe is part of our mostly it's part of our relationship with the sun. Deuterium may alternate from time to time based on oceanic temperatures, and we believe that we would be better off if, like in ancient times, deuterium would be lower in the environment. And now I'm considering some industrial processes that definitely enrich deuterium in food products, and we believe that these are also part of epidemiological scale of human diseases like diabetes, cancer, obesity, and some degenerative diseases. We believe that deuterium has a big role in these.
Absolutely. And I think that's a perfect segue. So if we summarize that, the environmental conditions, things like climate change, warming of the oceans have resulted in a level now that's higher than our ancestors would have been exposed to. Now, if we segue onto the food products, are you able to give us a bit of advice on what foods are higher in deuterium and which ones could perhaps be lower?
Yes, indeed. And this is, again, a very interesting topic because it seems like nature figured it out. Because in nature, so there are two basic type of food or substrates that we burn or oxidize in our mitochondria. One is called a carbohydrate, which, as its name says, it has carbons dissolved in water. Water exists in hydrogen and hydroxyl groups, or OH groups, and this is what, for example, glucose is. It has 6 carbons and 12 hydrogen, 6 oxygen, so it's practically 6 carbons dissolved in 6 water molecules in a chemical form, meaning that the hydrogens and the hydroxyl groups are attached to carbons, and this is typically a carbohydrate. On the other hand, fat is more of a hydrocarbon, meaning that there is no water in it. It's just only carbons and hydrogen. Mm-hmm. And those are the best source of hydrogen simply because fat or these hydrocarbons can only be produced from citric acid, which is part of mitochondrial metabolism. So because mitochondria can deplete deuterium, by filtering it out using these protein nanomotors, biological systems produce their own fat from one of the metabolites that can only be synthesized in mitochondria called citrate, and the enzyme citrate synthase is a mitochondrial bound enzyme. So there's no really other way of producing fat, most in the most robust pathways de novo from carbon and hydrogen than using mitochondrial deuterium depletion. And for that reason, natural fat, grass-fed animals fat, is low in deuterium. Carbohydrates, proteins depend on various other biochemical reactions, but those are usually high in deuterium. So carbohydrate diet may contain about 150, 140, 150, maybe in the higher end of 140s ppm, yet a natural ketogenic diet would only contain like 115 or 110 parts per million of hydrogen, deuterium. So that for deuterium burning, making metabolic water from fat in mitochondria is a lot more efficient, and it's a lot more beneficial for our cells because it's low in deuterium, and it gives you the highest rate of carbon to hydrogen, which powers our ATP synthase nanomotors the most efficiently in molar turnarounds of the TCA or Krebs-Sanger cycle. Yep. And this is what we call a ketogenic diet or natural ketogenic diet, when you consume mostly fat and you kind of use the benefits of these very basic fundamental biological biochemical principles.
Sure. So I'm hearing ketogenic is perhaps a better strategy in terms of deuterium depletion than say maybe a diet that's high in carbohydrates. How about fruits?
Again, let me add just one sentence here. It has to be grass-fed or natural.
Oh, yep, sure.
Like naturally produced fat. And the reason for that is it's very important that the deuterium discrimination process starts in grass. So photosynthesis, because the photons or the electromagnetic resonance that is carried in light, especially in red light, cannot break the oxygen deuterium bond. Therefore, photosynthesis discriminates against deuterium, because as we know, photosynthesis is practically breaking water from the ground that is soaked up by the tree leaves, and the tree leaves break this hydrogen and oxygen bond in water, and the hydrogen will be attached to carbon dioxide, and the oxygen is released. And this is why the trees produce oxygen and use carbon dioxide. So actually the circle of life starts with this very robust deuterium discrimination process in plants, and animal fat that we burn back into metabolic water after we ate them with the use of oxygen. Again, this is why I was saying this is like a circle, biochemical circle, magical circle. So that's practically the best way of producing low deuterium water that is produced or discriminated against, or the heavy water is discriminated against light water in plants. And because, and this would be the best way to answer a question, because fruits, on the other hand, are synthesized or produced by trees and plants using other biochemical reactions. They mostly use glucose or sugars to kind of wrap these very important like seed material of the trees that is for their propagation or propagating the species or kind of establishing new faunas through the seeds. That's practically carried in the belly of animals after eating the fruit. And it has to be high deuterium, so it's not going to be absorbed. It's going to be filtered out by the gut flora or the gut bacteria. And with that, the animals would have more like a diarrhea, and after that they would actually spread the seeds. And plants purposely produce high deuterium fruits simply to spread their seeds, because high deuterium sugars, high deuterium food, or high deuterium content proteins are not very efficiently absorbed into our circulation, simply because bacterial gut flora and also because of the transport processes are affected by deuterium. It seems that high deuterium sugars, they like to stay in the bowels, and after all, deuteria can munch on them. And for that reason, fruits and high fructose, which is fruit sugar, high fructose nutrients in that form, like high fructose corn syrup and so on, are not very desirable for human consumption, we believe.
Sure, and this is the Gut Health Gurus podcast, so I wouldn't be doing the podcast justice if I didn't go back to that exploration of the gut bacteria. Now, what's the effect of high deuterium levels or deuterium levels in general on your gut microbiome?
Yeah, so and that goes back again to a very fundamental biological principle called the prokaryotes. Those are bacteria, those are actually life forms, usually single life forms, which cannot control their deuterium content, or actually they retain deuterium by spinning these protein nanomotors to the opposite direction. And actually that was the first paper in 1988 published in Nature by scientists from Italy. They showed that in bacteria, in yeast, to be more precise, these nanomotors spin in a direction that retains deuterium for these yeast cells, and as a result of that, their DNA is constantly contaminated with deuterium, and for that reason they grow constantly. This is the prokaryotic life, and this is what's in the gut flora practically. These bacteria, they use deuterium and filter. They do us a great favor simply because they filter out deuterium from our food, but for that, you know, in the meantime, they keep growing constantly, and that's how stool becomes almost. I'm just, you know, I'm a doctor in that sense, so I want to explain this so you understand. So stool looks pretty much similar of like, you know, it's not really dependent what or how much you eat. Primarily, it's just practically bacteria and high deuterium food products, yet low deuterium fatty acids and triglycerides and so on, after digestive enzyme activities, they actually get absorbed in our circulation. And low deuterium food usually, or ketogenic diets usually decrease the bacterial amounts, and so that it is less of a stool production. But with high fructose, high glucose, high deuterium foods, carbohydrate, and flour and bread and grains, so on, you can actually increase, or fibers, you can actually increase, you know, the number of bacteria in your gut. But those are prokaryotes, so they retain deuterium to grow constantly, doing us a great favor to the eukaryote cells, which have actually mitochondria that can deplete deuterium in water, and their DNA is less contaminated with deuterium, and so that they can actually wrap it up in a cell nucleus, meaning that those DNA particles, compartments can rest for a while the cytoplasma or the cell is differentiating or delivering some more sophisticated functions, and more precise, and more we call them differentiation, when cells become more and more precise in their function, instead of dividing constantly like bacteria do. And this is a big difference between prokaryote and eukaryote cells, that eukaryotes, because they can control their deuterium with the help of prokaryotes in that sense, which actually absorb deuterium or process deuterium in the gut. And this is why your podcast is so interesting, because we are just learning this new function of deuterium on gut flora, and I think this will be very critical in understanding what the gut flora does after all for us. But it seems that you know, the deuterium explains the two major cellular phenotypes as far as prokaryotes, just like bacteria, and eukaryotes, just like human, like liver cells, do practically the size of their DNA is different, and for that reason they can specialize if they are less saturated with deuterium. And this role, this very important, you know, kind of switching or splitting duties, is between the gut flora and the rest of our body. Practically, the gut flora will normalize or try to process deuterium in the gut before it gets absorbed. And obviously, if there's too much in food, if there's too much deuterium in the crops that we eat, or in the food that we consume, or the routines that we elaborate around healthy eating or eating in general, deuterium, after all, would be just too much for the bacterial gut flora to deplete it completely. So our body can be more and more saturated with the disease processes appearing in that metabolic or degenerative or cellular proliferative, or even our human cells can become bacterial or prokaryote phenotype, in that sense that you know, they accumulate deuterium and start going in an uncontrolled fashion in the form of cancer, for example.
Sure. Now, we're pretty much coming up to time, Dr. Laszlo. There's just a couple more questions that I'd really like to get through. And firstly, is there a level of deuterium where it could be damaging to prokaryotes?
Prokaryotes, we don't know necessarily how far they can go as like very high deuterium concentrations. We believe that anything above 165, 170 ppm is very damaging to eukaryote cells, but for prokaryotes, I don't think there's an upper limit. Yet they actually waste deuterium back into the soil or back into the environment simply because they are not stable life forms. So rapid growth is always followed by rapid death.
Sure.
So usually bacterial cultures or yeast are famous of making things rotten or producing pus or after all dismantling themselves because of the high deuterium saturation. With low deuterium contents, we know below 40 ppm, like nothing would grow. Even prokaryotes slow down significantly their growth rates. And the lower limits are much more kind of precisely defined than the high range, especially when it comes to prokaryotes. But this is definitely a very important biological information of how we distribute deuterium among different cellular forms and cellular types, and how we split the duty of controlling, deuterium content in our cells, deplete as much as possible when it comes to mitochondrial functions. And the full understanding of these systems start from knowing these very basic life forms, prokaryotes and eukaryotes, and it seems that it works very well it has worked very well for biology for long times, and the future is questionable simply because we have now epidemiological population scale disease processes, which we
Sure.
believe, after all, would be much better handled once deuterium biochemistry or deuterium physiology or deuterium-based medicine is taking roles.
Absolutely. And there's a couple of, additional strategies I'd like to explore in terms of deuterium depletion. Breathing. Now, how does breathing affect deuterium?
So by breathing, you carry oxygen to your tissues, and your breath is deuterium depleted in the first place. So humans, by gathering and breathing fume or their own vapor, depletes deuterium after all, simply because human breath, if it's a healthy individual, it's probably in the range of 140, 135 ppm. And simply because our metabolic water, which come from our tissue and being exhaled through breath, through breathing, is lower in deuterium, as we mentioned. So this is measurable. So this is easy to measure, these biological principles. And by breathing, by decreasing oxygen tension somewhat, can actually dilate your vessels. And as a result of that, there is more efficient oxygen delivery, especially when there's increasing carbon dioxide partial pressure. So breathing controls metabolic water production through dissolved oxygen in your plasma, oxygen that is bound to hemoglobin and carried to cells, to the mitochondria, and also by carbon dioxide, which is released as a result of biological substrate oxidation. And these factors, they all control metabolic water production with some very important hormones like ADH, which is an antidiuretic hormone that retains water in our cells or in our body. If we don't dilute it, if we don't drink external water, too much external water, especially when we are not thirsty, it's really useless to drink so much water. Breathing is much more important, and light is very important, because light, especially red light, can resonate the oxygen, hydrogen, and carbon-hydrogen bonds. Red light makes more water more viscous, easier to flow in our mitochondria. Mitochondria has this surface or tension water that has to be controlled by its viscosity to regulate cellular functions, and red light actually helps our cells, our mitochondria, and also carbon-hydrogen resonance in the form of cholesterol and fatty acid, free fatty acid molecules or triglycerides are also very important to kind of resonate with light and communicate energy with our cells through these hydrogen bonds. And deuterium compromises these functions of both light, oxygen, and carbon dioxide. So for that reason, this is just an additional physiological level that hydrogen deuterium physiology comes very handy. Deuterium does not resonate efficiently. Deuterium does not absorb the energy of light. So this is why deuterium depletion is also important. And also breathing is very important. Light and breathing are very important to maintain health.
Absolutely fascinating. And I think just one final loop. Now, you mentioned light. Now, with light and something that we were discussing earlier with light and sleep and melatonin. Now, how are these three individual components involved in deuterium depletion?
Yeah, so it seems that sleep is practically a deuterium depleting process during burning fat at night using mitochondria. Our melatonin is released in dark, just in lay terms. So light exposure usually decreases melatonin release, yet when it gets dark, our melatonin gets high and it accumulates in mitochondria and peroxisomes. And for that reason, when you fall asleep, then you very quickly use up your glucose in your circulation. If you were awake, you would be hungry and you would eat, but nature took care of this, so you actually felt unconscious while you're sleeping, so you don't feel your hunger. So you allow your body and your melatonin to oxidize fat, low deuterium fat, to produce metabolic water during night sleep to deplete your deuterium that you accumulated when you were awake. So for that reason, it seems that the light cycle regulates this deuterium depletion process simply using hormones that activate in our mitochondria and our peroxisomes water-producing enzymes or water-producing biochemical architecture or a number of enzymatic steps. After all, in the morning time when we wake up, we have less deuterium in our circulation. We have burned fat during night sleep efficiently in peroxisomes, which is low deuterium, which results in low deuterium metabolic water. So we are ready to go again until, in the light cycle, until it gets dark again and we start the process all over.
Wow, and that's absolutely fascinating. And we'll link to, I guess, the article that you sent me earlier today about the impact of light on melatonin and potential links to breast cancer. Do you want to talk very quickly about that?
Yeah, so Endocrinology 2019, the conference features a talk this year from the University of New Orleans Medical School. And it shows that actually if there is light during sleep, many people like just to leave lights on. It actually compromises melatonin secretion. And the result of that, in a cancer model, scientists were able to show that breast cancer injected in mice metastasizes through the bones much more rapidly when there is light in the cycle and continuous light exposure, even at night, at very low dim or very low light sources is present. The behavior, the biological behavior of breast cancer in this animal model would dramatically change. And I would encourage people to look at the Endocrinology 2019 news releases, because this information is so linking light and melatonin with cancer progression through bone metastasis, and also adding deuterium and understanding of how the system works from our side. Listening to this podcast, which is an honor to me, would explain how and why this model turned out to be a light-related cancer propagating or cancer growth promoting process. Yet, if we follow the natural light patterns, meaning that we sleep in dark and allow our melatonin to hit high concentrations in our mitochondria and liposomes, then we may need less time to sleep. We are more rested when we wake up, and we depleted more deuterium more efficiently. So we preserved our health during these very interconnected, very fundamental biological processes. And this is what is demonstrated in this abstract, and it's absolutely a fascinating study to linking light, melatonin, and cancer growth. And I'm very happy we were able to add our biochemical interpretations to this, and obviously with the help of your podcast to explain this to some very kind audience and people who are interested in the topic.
Sure, and we'll definitely link to the article in the show notes. And just one final question: deuterium depleted water. Now, I think you kind of started to talk about the process of removing or mechanically removing deuterium, or there's some sort of chemical process to remove deuterium from water and get it down to around 25 ppm. Now, in what situation would people be needing to, I guess, use a type of water that's, I guess, that depleted?
Yeah, so my advice, or my teaching curriculum is focused on water simply for its deuterium content, and this is a very good question, simply because this relates to how much water we should drink and when we should drink water. Mm-hmm. In healthy individuals, usually, and this is what I recommend to drink when you're thirsty, and wait a little bit and you're still thirsty, because antidiuretic hormone is very important in many physiological processes. So for healthy individuals, drinking rainwater, to that matter, which is somewhat deuterium depleted, or drinking deuterium depleted water around that, or somewhere in the surroundings of 120 ppm or below, that's desirable. If somebody has a disease, for example, cancer, diabetes, or has obesity, and they want quick start on deuterium depletion, then we recommend them, or we suggest, to learn about how lower deuterium content, like 85 ppm or even 50 or 40 ppm deuterium depleted water, may get them on a quick start and start depleting deuterium very quickly to slow down cancer growth, for example, or maybe get the glucose or metabolism in a better shape, glucose status. So we use this ketogenic diet, deuterium depleting ketogenic diet, now to even treat type 1 diabetes. So wow. It has a dramatic effect on, the American Pediatrics Association, just the paper, the Journal of the American Pediatrics Association called Pediatrics has just published a paper treating type 1 diabetes successfully using ketogenic diets. And my colleagues in Hungary, Dr. Csabatóth and Zsófia Clemens, you may want to talk to them sometime in the future, but definitely they use ketogenic natural diets, low deuterium ketogenic diets. They call them paleolithic or evolutionary medicine, to treat a number of disease conditions, and they are clinicians. They can elaborate, and they publish in the field. So there are case reports which show the beneficial effects of deuterium depletion. But as you said, mostly cellular proliferative, like cancer diseases, and also metabolic diseases like obesity and the metabolic syndrome. They all benefit from a ketogenic, low deuterium ketogenic diet, and also consuming deuterium depleted water in like low ranges, like 85 to like 40 ppm.
Sure. And Dr. Laszlo, just because most of our audience are going to be Australian, we do get a lot of US listeners as well. But particularly for Australian audiences, now our deuterium level in our drinking water, in our rainwater, I would say is in a higher end of the scale. So if seawater was 155, our drinking water is 153 ppm. So what's your take on that? What
Yeah, well, be honest with you, it's not appropriate for human consumption.
Wow. And that's
You
just
know, you are in a very dangerous zone
yeah,
in that
absolutely.
sense.
Absolutely.
So what Australians should do is eat more beef, more pork fat, or more like meat that is high in fat, and produce their own water in their mitochondria instead of drinking water like bottled water that is sold in stores, and wait
Yeah.
till they get thirsty and don't drink just because somebody told them to drink based on you know, economically motivated suggestions.
Mm-hmm.
You should, I think, biochemically, you should learn. Again, we are a teaching facility. I teach biochemistry of deuterium. So my recommendation is to produce your own water when you don't trust, or can't trust your external water. Your mitochondria are perfect to produce the optimal amount of the best deuterium depleted water for your physiological and biochemical reactions and health, to that matter. And for that reason, is just stay away from from water, bottled water that is very high in deuterium. I think that's a
Sure. And for
fair
us,
statement. You should
yeah,
learn about this by exploring additional information. What you hear in this podcast, I'm just covering the very basics, but practically, like in the ballpark range, you should not drink water even when you're thirsty, anything above like 100, like 35 ppm, which is rainwater. And that's practically the cleanest, and that's the lowest deuterium as far as external water goes, because that's fractionated. And Australia, unfortunately, because the island is in the middle of the Pacific, middle of the in the ocean, your external water will be, through the vapor and the evaporation, it will be high in deuterium,
yes.
unless there are high elevations in Australia, which I don't think. Do you guys have big mountains?
We do have mountains, but certainly not like in Europe when you've got these, or in other regions where you've got the glacial type waters. Our water, from what we've tested, is
Yeah. We
high.
we know about this problem
Yeah.
in Australia. So just keep eating and keep producing your own water. I think that's the safest.
Yeah, and what we'll do is we'll certainly link to the Deuterium Depletion Centre as well, which you're part of, because I think it's an amazing resource, and particularly for people in Australia and in New Zealand and island type nations that don't have the luxury of having rainwater at 135, where we need help. I mean, you can see it in the rates of cancers and obesity and illness in Australia. We need your help, Dr. Boros. So I'll definitely be linking to your website.
Yeah, thank you so much. Yet again, one of our colleagues, Dr. Cooper, is from Australia. So, and she is now definitely one of the experts, clinicians in deuterium depletion. We are very happy to help or study this question or this problem further with our Australian friends and colleagues. We are there for you guys anytime you need us, and I hope we can give you some very useful and beneficial advice based on biochemistry teaching, and you can actually go after this information by yourself, as we point to the source of literature or published studies that we use for our own interpretations. We definitely have studied this for over a longer period of time, and we teach this accordingly, and hopefully this type of information will be helpful to our friends and our colleagues in Australia.
Absolutely. And Dr. Laszlo, one final question before we start to wrap. What if there was one thing that you would recommend for our audience to improve their health? It could be gut health, mitochondrial health, any health. What would be your top tip?
It's practically listen to your own body as far as hunger and thirst and what to desire to eat, with the knowledge of how to sense deuterium in the environment. Animals can differentiate among water that high in deuterium. Now we also can, as we have studied. It was funny because I was in China and they gave us blindly water with deuterium depletion, I think 105 tap water and 50 ppm, and we picked them all correctly several times blindly mixing. Because you have to learn. I think humans lost their ability to kind of sense viscosity of water or acidity of water that is controlled by protons and deuterium. But you can learn this very quick, and you can actually guide yourself in your environment what you should or what you shouldn't drink. We can't really, by taste or by smoothness of water, you can actually learn how to differentiate among water sources with different deuterium concentrations. Again, you have to learn about this process, and you have to kind of live with it, or have to kind of live a lifestyle that is more like deuterium conscious. Yet, I think after all, if you eat what you desire, it's mostly ketogenic, like fat and meat based, with some vegetables and some greens that are also grown naturally, excluding industrial food processes or processed food. And you drink deuterium depleted water when you're thirsty, and you also drink as much as you need to kill your thirst without supplements, because those are really not necessary unless you need it medically. Unless you're deficient, the best way to handle your mitochondrial health is to be exposed to light as much as possible in your local growths and local environments, to consume food that is local, mostly fat-based, mostly ketogenic, and drink very little, if necessary, water, and allow your body to produce its own water and retain its own water by the combination of antidiuretic hormone, light exposure, melatonin, sleep, good sleep in the dark, not to be exposed to electromagnetic waves, including lead, and so on. Everything that Dr. Jack Kruse and Dr. Gabor Somlyai were mentioning with my biochemistry background and teaching are very important, after all, to maintain your health in a good state, and obviously this is the main goal of our activities at the centers of deuterium depletion, to bring this knowledge and some experience to people.
Wow, amazing. And what a beautiful way to finish the podcast. I just want to thank you for bringing this revolutionary information to our audience here in Australia and across the world. Thank you so much, Dr. Laszlo Boros.
Thank you. And if you have questions or you want to follow up, if you connect with our centers, then you can explore this further for your health and good life.
Wonderful. Thank you, Dr. Laszlo.